guys, welcome back to the podcast. On today's episode, we are going to be talking about synthetic biology improving upon nature. So, with the growing world, the human population is seeing more and more cases of disease, causing serious life-threatening and life-changing symptoms. Plants contain a great untapped source of bioactive chemicals with the potential to improve human health through the prevention as well as treatment of disease. Synthetic plant biology is an ever-changing, ever-improving, and ever-inventing field that can use the power of chemistry in plants to improve the production of these known and unknown therapeutic substances, which would allow us to increase our ability to produce complex pharmaceuticals in plant systems. But how do we synthesize them in large quantities for therapeutic purposes? How do we do this? Transient gene expression is the answer. The big picture here, before we get into the nitty-gritty, is to be able to target a compound, understand the metabolic pathway that brings this compound to life, and then enhance the pathway in a way that increases the production of the compound inside the plant. By doing this, scientists and researchers will be able to produce large amounts of extremely complex compounds that have known and unknown therapeutic properties to treat diseases that have plagued the human race since its origin. Transient gene expression is at the forefront of research right now, and for a very, very good reason. Agrobacterium-mediated transient transformation is a technique to get transient and high-level expression of target genes, which is facile and versatile for the characterization of gene function in plants. This transient expression is a recently developed approach to quick and inexpensive large-scale production of recombinant proteins in plant systems. The process uses the microbe Agrobacterium, which is the only known bacterium to be able to transfer genes to plants. In the course of this process, foreign gene expression occurs during several days after agroinfiltration without integration of recombinant DNA into the plant genome, and the level of target protein synthesis, so the compound that these researchers want to increase the production of, may be considerably higher than in stably transformed plants. This technique only requires a wait time of 3 to 6 days to see the expression and characterization of the desired gene in the host plant. This is extremely comparable to the months it would take to grow stable plant lines heterologously expressed in a greenhouse with a very, very sad graduate student, and I would know because I worked with multiple on the second floor of ATRB and in their greenhouse, and I think about them often. So in this process, there are quite a few moving parts, but the main point is that Agrobacterium is modified to highly express a gene that researchers want to amplify in the host plant. So circling back to the point, as I may have lost you guys, our main focus is the improvement of therapeutic compound production in plants that may be in low abundance or low cultivation. So this transient gene expression would increase these compounds' production, allowing us to use them in medicinal advancements. So researchers found a solution to solve the problems of low abundance using heterologous expression of these bioactive compounds for large-scale production and use, and that solution is transient gene expression. Modern sequencing technologies have also become so advanced that whole genome sequencing and transcriptomics in concomitance with co-expression analysis, phylogenomics, gene clustering, and genome-wide association studies of non-model species have become accessible. Now, why is this important? You're like, you just said a whole bunch of nothing to me. So this is important is because the techniques open the door for rapid and quick discovery of every little factory station in complex biosynthetic pathways, thus resulting in the ability to provoke the heterologous expression of desired pathways for specific compound production. As we come to the conclusion of this podcast, I want you to remember its purpose. Our topic today was the use of synthetic biology to improve upon nature. We discussed the field of synthetic biology and its importance in the multitude of discoveries of specialized metabolites and compounds that may very well hold the key to treating many chronic and deadly diseases that plague the human race. We focused on the ongoing research being performed to explore and voyage through the complex chemical pathways used by plants to produce these compounds, and discussed the methods being used to enhance and increase the production of low abundance compounds. Plants contain a great untapped source of bioactive chemicals with the potential to improve human health for the prevention as well as treatment of disease. As long as the plants grow, we will continue to discover, learn, and change the world.